In this video, we will look at the normal structure and function of the hepatobiliary and pancreatic systems. The pancreas and liver are two of the major accessory organs associated with the GI tract. The pancreas lies posterior to the stomach, and it runs from the duodenum over toward the spleen. It is bound to the posterior wall of the abdominal cavity and wrapped in a thin connective tissue capsule. The pancreas has three major regions to it. The first is the head. This is the broadest section of the pancreas, found nestled in the loop of the duodenum. This is also how the pancreas is attached to the duodenum via the pancreatic duct. The body is the long, tapered, slender portion that extends toward the spleen. And finally, the tail is the short, rounded tip. When you look at a histological slide of the pancreas, you will notice two distinct types of cells. The majority of the cells in the pancreas are arranged as lobules. Each lobule consists of blood vessels and tributaries of pancreatic ducts. Each of these lobules will then end in a blind pocket known as a pancreatic acini. Here in this top picture, we see the pancreatic duct coming down into its tributaries and then ending in these little blind pockets called the acini. The other type of cell in the pancreas are pancreatic islands. These only account for about 1% of all of the pancreatic cells. The pancreatic islets represent the endocrine portion of the pancreas, and we can see one of these islets right here as well as right here in the upper picture. The pancreas serves two main functions. It acts both as an endocrine organ as well as an exocrine organ. The endocrine cells of the pancreas are found within the pancreatic islets. They secrete two major hormones, insulin and glucagon, into the bloodstream. We will talk more about these when we cover the endocrine chapter. The exocrine portion consists of the acinar cells and the epithelial cells of the duct system. Their job is to produce the various components of pancreatic juice. These pancreatic secretions, also known as pancreatic juice, consists of numerous enzymes. One of these is pancreatic alpha amylase. This enzyme breaks down starches. It is very similar to salivary amylase that is found in the mouth. Another enzyme is pancreatic lipase. This one breaks down complex lipids into smaller products that are more easily absorbed. You will also find nucleases. Their job is to break down nucleic acids. And then about 70% of all of the enzymes found in pancreatic juice are proteolytic enzymes. Their job is to break down proteins. There are several different proteolytic enzymes, each of which will recognize a different portion or component of a protein. They are typically secreted as an inactive proenzyme and will not be activated until after they reach the intestine. Next is the liver. This is the largest visceral organ in the abdominal cavity, averaging about 3.3 pounds. It is found in the right hypochondriac and epigastric region, and then extends to the left hypochondriac and umbilical region. The liver is responsible for performing numerous metabolic and synthetic functions for the body. Looking at the gross structure, we see that the liver is divided into lobes. Two of the major lobes are seen here, the right lobe and the left lobe. The liver is then wrapped in a tough fibrous capsule and covered by a thin visceral peritoneum. The lobes are separated by ligaments, two of which can be seen here. The largest ligament is the falciform ligament that is found between the right and the left lobe. This acts as a major anatomical landmark, especially for surgeons. And then on the top, we see the coronary ligament. This helps to anchor the liver to the diaphragm. When we look at a histological preparation of the liver, we notice that it too is organized in the form of lobules. The liver lobule is a basic functional unit of the liver. It appears in the shape of a hexagon. At each of the points of these hexagons, you will find a portal area, also called a hepatic triad. Within this portal area, you will find three structures. One of these structures is the hepatic portal vein, a branch of the hepatic portal vein. This is the blue structure. You will also find a branch of the hepatic artery, and lastly, a small branch of the bile duct. The majority of the cells in the liver are known as hepatocytes. These form a series of irregular plates that are arranged like the spokes of a wheel. Interspersed within the sinusoids between these hepatocytes, you may also find Kupffer cells. These are an example of fixed macrophages. We do not see any highlighted in this particular picture. As blood enters the liver, it will flow through the sinusoids. As it does so, the hepatocytes will absorb solutes and nutrients from the plasma. The hepatocytes will also secrete various materials such as plasma proteins and glucose into the blood. One of the main functions of the liver is to produce and secrete bile. The liver will secrete this bile into a network of narrow channels called bile canaliculi. These will be found between opposing membranes of adjacent liver cells. We can see some of these fine green finger-like structures known as the canaliculi here in this picture. All of these bile canaliculi will drain into small bile ducts. Then all of these small bile ducts come together, forming one of two major ducts, either the right or the left hepatic duct. It is the job of these two major ducts to collect all of the bile from all of the bile ducts in the liver lobes. The right and left hepatic duct will then unite to form the common hepatic duct that leaves the liver. Again, we can see this here. Here are the right and left hepatic ducts, and they join to become the common hepatic duct. When bile is produced and secreted, it will leave the liver through the common hepatic duct. Once in the common hepatic duct, the bile has one of two choices as far as where to flow next. It can go from the common hepatic duct into the common bile duct, which empties into the duodenum. Or it can enter into the cystic duct, which heads up to the gallbladder, which will then store the bile until it is needed. So again, here we see the common hepatic duct. It can go into the common bile duct, which will drain into the duodenum, or it can go into the cystic duct, which drains into the gallbladder. The common bile duct is formed by the union of the cystic duct, which is coming out of the gallbladder, and the common hepatic duct, which leads the liver. The common bile duct will penetrate the wall of the duodenum. It actually meets up with the pancreatic duct coming from the pancreas at the same duodenal ampulla. So here we see the common bile duct meeting up with the pancreatic duct and draining through the ampule into the duodenum. Here we see the common hepatic duct and the cystic duct coming together to make the common bile duct. The liver has various physiological functions, or performs various physiological functions for the body. The first of these is metabolic regulation. The liver is responsible for regulating the composition of circulating blood. It will regulate nutrient metabolism, waste product removal from the blood, nutrient storage, and drug inactivation. The liver is also involved in hematological regulation. The liver represents the largest blood reservoir in the body, receiving upwards of 25% of the cardiac output. Some of these hematological functions include phagocytosis and antigen presentation to help the body mount an immune response. The liver will synthesize plasma proteins and put them into the blood. It will remove circulating hormones so that they do not achieve too high of a level in the blood. It will remove antibodies, be it either excess antibodies or spent antibodies. It will remove or store toxins and synthesize and secrete bile. We looked before at how bile flows through the liver, but what is the actual function of bile? Dietary lipids are not water soluble. As these lipids are processed in the stomach via mechanical processing, this creates large drops of lipids, which are very difficult for the body to process further. When these enter into the duodenum, bile salts will mix with them. The bile salts will break these droplets apart into smaller components. This is known as emulsification. This will increase the surface area exposed to enzymatic attack and create these tiny emulsion droplets coated with bile salts that are much easier for the body to process. The last structure to look at is the gallbladder. This is a pear-shaped muscular sac located in the fossa on the posterior surface of the liver's right lobe. The function of the gallbladder is to store and concentrate the bile prior to its excretion into the small intestines. The function of the gallbladder is to store this bile until it is needed. The gallbladder will release bile into the duodenum when it receives the stimulation of the hormone cholecystokinin, also called CCK. A full gallbladder contains anywhere from 40 to 70 milliliters of bile. The composition of the bile gradually changes as it is stored in the gallbladder. This is mainly due to the fact that water will be reabsorbed as it's stored. As this water is reabsorbed, the bile salts and the solutes in the bile will become more concentrated.